Thank you very much. Let me first thank Angelique Botha for inviting me to contribute to this webinar, and Steve Allison for all the arrangements that he has kindly made. I also wish to thank Stephanie Trapman for welcoming me, contributing to ISO 33406 that she described earlier today, and to congratulate her for her exemplary leadership that led to its successful conclusion. And I thank all of you, the attendees today, for your interest in this webinar in general and for staying for my presentation that I will next show. I will review two examples of NIST reference materials with certified values of nominal properties. They were issued before the publication of the ISO standard that I just mentioned, 33406. The first is Standard Reference Material 3246, originally certified in 2007, comprising powdered leaves of Ginkgo biloba, the maidenhair tree, showed here in this picture, with the beautiful leaves of fall in yellow and the green leaves of spring and summer. The second is reference material 8256, wild-caught coho salmon, originally certified in 2021, comprising tissue of this species caught in the Gulf of Alaska. I was personally involved in the development of the second, but not of the first. Here's the top of the first page of the certificate for SRM 3246, stating its purpose, the properties, whose values it delivers, and listing the institutions that collaborated in its development. Ginkgo was widely believed to have been extinguished since about 3 million years ago, showing up in the fossil record as early as the time of the dinosaurs. However, a German botanist found living ginkgos in China in 1690, and nowadays they are cultivated the world over. Ginkgo biloba is an exceptional living being. Its genome is about 3 times longer than the human genome. It produces a wealth of complex phytochemicals and defies the common effects of aging, even among trees. 6 ginkgos within 2 kilometers of the detonation site of Little Boy over Hiroshima survived the blast, and soon thereafter were healthy again and are still living. The certified property values for the ginkgo are the identity of individual nucleotides in its DNA, the biological species, based on taxonomy, genome, and phylogenesis, and mass fractions of flavonoids, ginkolide B, cadmium, lead, and mercury. The confidence evaluations made against an ordinal scale of levels of confidence yield most confident, both for DNA nucleotide identity and for species identity. For the nucleotide identity, this means that we have reliable, unambiguous base calls on both strands, and that all data from both strands agree. For the species identity, this means that the phylogeny is well supported and well resolved. There are multiple diagnostic sequences of nucleotides differentiating the species from closest relatives, and we have data from multiple samples in both inclusivity and exclusivity panels. Finally, the data are mutually consistent from several regions of the genome. Sanger sequencing of both DNA strands of 2 particular regions of the genome supported the most confident identification of the botanical species that is the source of the material. Validation studies based on a probability of identification statistical model, including inclusivity and exclusivity panels, confirmed the species identification. These studies involved analyzing analyzing samples of authenticated herbarium vouchers and of botanical identity reference materials the same way as the material from these SRM 3246 was analyzed. Maximum likelihood phylogenetic analysis using the PhiML algorithm show that these material clusters very closely with authenticated exemplars of Ginkgo biloba. They're all in green in the diagram on the right. And being phylogenetically distant from similar species in other genera, for example, Cycas, which is depicted down at the bottom. Next, I will turn to reference material 8256, fresh frozen tissue of coho salmon, supporting investigations of seafood safety and seafood authenticity. Coho is one of the 6 species of Pacific salmon, reaching a mass of 16 kilograms in a 5-year-long lifespan. This fish is born in a river, spends most of its lifetime in the Pacific Ocean, and returns to the same river where it was born to spawn and then to die. Similarly to many other species of fish, coho can be identified using a sequence about 650 nucleobases long from the cytochrome C oxidase subunit 1 of the mitochondrial gene COI, or CO1, Roman letter, or a Roman numeral at the end. The material was cryo-homogenized prior to bottling, and the homogeneity of the contents of the bottles was evaluated from chemical and genetic viewpoints. Total crude fat determinations in a sample of jars had 4% relative standard deviation, no significant bias, and no significant trend as function of bottle number. And relative standard uncertainties of mass fractions of arsenic, copper, iron, manganese, selenium, and zinc in a sample of jars ranged from about 2% to 8%. The genetic homogeneity was tested and evaluated by a partner laboratory, NSF International, and revealed that about 99.993% with standard uncertainty 0.004% of the DNA sequences in the material are from Oncorhynchus kisuch, which is the coho salmon. NIST is most confident in the identity of this material, which was achieved based on 3 identification criteria that I will review next. The first criterion was a phylogeny reconstruction based on sequences regions CO3 and ND3 of mitochondrial DNA and comparing them against reference sequences from NOAA's Northwest Fisheries Science Center. The second criterion involved comparing nucleobase sequences from the same regions, CO3 and ND3, of the same mitochondrial DNA against reference sequences of authenticated salmon, coho salmon, to establish inclusivity, and against similar sequences from close relatives in the family of the salmonids to establish exclusivity. The third criterion involved next-gen sequencing of mitochondrial DNA fragments and Monte Carlo perturbations to express nucleotide identification error probabilities, of which I'll show you a diagram next. Then alignment and comparison against reference sequences, often called genetic barcodes, of voucher specimens of different species of Pacific salmon and other similar species that are stored in the reference standard sequence library of the US Food and Drug Administration and also from the Barcode of Life data system. The idea of using barcodes like these goes back to Herbert and his co-authors in a paper they published in 2003, motivated by the realization that expertise in classical taxonomy was vanishing. They regarded the use of DNA barcodes as the sole prospect for a sustainable identification capability going forward. And indeed, their forecast came very much true. Let me say a few words about the uncertainty evaluation surrounding the nucleobase identity. I'll show you a diagram, as I said, in just a second, but let me first tell you in words what was done. One particular locus was identified as adenine, and qualified with a quality score that can be translated into an error probability. All of these sequences, these short reads, were stored in a FASTQ file that has not only the sequences, but also the quality scores that go locus by locus together with the sequences. For this particular locus, the error probability was unusually high, 16%. So what one does, absent other information, is that the nucleobase identification probabilities at this locus are 84% for adenine, and then 5.3% for each of the other three possibilities, cytosine, guanine, and thymine. Monte Carlo sampling of similar distributions for all the other loci in this DNA fragment generates replicates of fragments composition. And here is an example. Inside this rectangular box, you see a short read, and then a second line, which is a Monte Carlo replicate that differs from the other one in the sites indicated in red. And these came about by doing this Monte Carlo exercise that I just described for you. In addition to that, once we have a Monte Carlo replicate that reflects the uncertainty in the nucleotide identification, we produce an alignment against the barcode. So in this case, here's the barcode at the bottom, 650 so many nucleobases for coho. Of course, we did it for several different species, but this is one of them. And here is the alignment that was produced by Robert Edgar's muscle, version 5.2. Robert Edgar is a prime example of a gentleman scholar. He is unaffiliated with any institution, and he produces remarkable work in molecular genetics, which he publishes in very reputable journals and makes these codes, many of these codes, available for free. For each Monte Carlo replicate, an ensemble of high accuracy alignments is produced, and this is just one of them. The two things together allow us to evaluate these two sources of uncertainty, nucleobase identification and alignment uncertainty. Here is a summary of the results of these third criterion for species identification. The results are summarized in terms of a probability distribution over species of Pacific salmon and closely related species. So here you have their common names. So coho, steelhead, chinook, sockeye, pink, and chum all are the conventional Pacific salmon, Pacific on our side of the Pacific. These are all species of the same genus, Oncorhynchus. The Atlantic salmon and the brown trout both belong to a different genus, Salmo. And the Sakhalin taimen, who is also a Pacific salmonid, but on the other side of the Pacific, on the Japanese and Russian side of the Pacific, and the Arctic char belong to two other genera of salmonids. And coho comes across as an overwhelming favorite in terms of confidence for the identification of this material. The brown trout is a surprising second best, but a very distant second best, with chinook and steelhead coming next, also very distant third and fourth choices. These 0.00% are not really 000 all the way to the end, but the significant digits that are not zero are beyond the second decimal in the percent scale. These results are based on 100 fragments of mitochondrial DNA from gene CO1, which is what I believe an amazing result, because there are thousands, many thousands of such short reads in the FASTQ file, and a mere sample of 100 can accomplish these very reliable identification. Not only that, these fragments were chosen to have between 100 and 200 nucleobases, which is about a third of the length of the barcode. So this statistical method of identification, based on very short reads, and a small number of them at that, achieves this remarkable confidence in identification between very closely related species and some other species that are not so close, but still close enough to make a difference in terms of judgment, the quality of the results. And this is the story that I have for you today. I thank you again for your attention and for the opportunity to present to you.